Happy Monday, everyone. How's everyone doing? How's life? Stressed? I feel you. I'm so tired. We're almost there. So I'm gonna pull up my second screen. So in terms of logistical announcements, I think nothing too special. You all had Lab 5 due last night at 10 p.m. So hopefully everyone got that in. And as always, the next lab is out, so Lab 6 is released, and it's gonna be due this upcoming Sunday at 10 p.m. So hopefully you've gotten into the groove of things. I think this is also the last week that folks that are taking the midterm, I think this is the final week to do it. So hopefully if you've registered for this week, don't forget about your exam time because you won't be able to reschedule any later because this is the final week. So yeah, kind of keep an eye on that if you haven't taken it already. And yeah, just 4 more labs and then the final, and then we're done. Home stretch. And then it's summer. Woo. Awesome. Okay, any questions about course logistics? Any concerns, comments, dreams? Yeah, go for it.
For the final, is it going to be everything comprehensive, or is it like after the midterm?
Good question. Yeah. So the question was, is the final going to be comprehensive or just after the midterm? So the final is going to be comprehensive, and let me pull up the syllabus. And I do also want to now that you mentioned it, I'm going to highlight this policy here, which we have because the final is comprehensive. So hopefully everyone does great on the midterm, and hopefully you studied up and you're going to do great. If you didn't do as hot as you were hoping, that's totally fine. Because the final is comprehensive, if you score higher on the final than you did on the midterm, we're going to upscale your final and downscale your midterm. So if you didn't do as great as you were hoping, just study up for the final and you'll have like a chance for redemption. Yeah. I can't switch it, sorry. But it's all good. If you did good on the midterm, that means you're going to do great on the final. There we go. Okay, yeah, great question though. Any other logistical questions, concerns, comments? Everyone's with me? 1 more month. We got this. 4 more assignments. Okay, so let me go ahead and pull this back over here. Let me open up my slides. Let's see what we got. Open up recordings. Open up, whoops. Oh, geez. Open up my slides. And then, okay, presenter view. Oops. Drag this over. Okie dokes. And hopefully folks on Zoom can probably still see. So hopefully that's the case. If not, then just yell at me and I will fix it. Cool. So as always, let's start with the daily recap. We're 6 weeks in, time flies. So what have we talked about so far? So week 1, we introduced all these cool problems we want to tackle. Week 2, we talked about de novo genome assembly and genome annotation. So given this unknown pathogen, how do we reconstruct its genome sequence? How do we annotate that genome sequence to see what are the genes? What do those genes do? Yada, yada. Week 3, we talked about read mapping. So now that we have a high confidence assembled genome sequence of this new pathogen, if we sequence new samples of this pathogen, how do we map those reads to the reference genome? And the thought process was every sample that I collect of this virus, it's all from that same species. So I expect there to be a little bit of deviation from that reference genome, but they're going to be almost identical. So comparing against the reference genome makes it much easier for us to do downstream analyses. What downstream analyses do we want to do? Week 4, we talked about variant calling and consensus sequence calling. So consensus sequence calling was, how do I actually reconstruct the entire consensus sequence of this sample? So basically, for each position of the genome, let me take the consensus nucleotide, the most frequent nucleotide, and just output it for the whole genome. That is my best guess of the genome sequence of the sample. And then we also talked about variant calling, where given that all of these sequences are almost identical, maybe we don't want to redundantly show the entire 99.9% identical sequence. Maybe we just show deltas with respect to the reference genome. So for viruses, usually we just show the whole genome sequence just because viral genomes are short. But if you do any like human genetics or anything like that, we usually report variants in the VCF format that just show how does this person differ from the reference genome. Then last week, we talked about, okay, now let's imagine the pandemic has been going on for a while. We've been sequencing genome sequences from viruses all over the world. What do we do with them? And we talked about a problem known as multiple sequence alignment. So when we talked about read mapping, we talked about pairwise sequence alignment, which is a way of lining up two sequences to figure out their sequence level homology. So basically, in the same column, you have the same letter descending from the same exact position of the ancestral genome sequence. How do I do that for a bunch of sequences? So we introduced multiple sequence alignment. We introduced the inefficient algorithm that would be exponential time. We talked about the faster heuristics that exist. And then we ended by talking about a tool that I wrote called Viral MSA that uses a reference genome to speed up the multiple sequence alignment problem using read mapping tools. So at the end of last week, we now have a multiple sequence alignment with like millions of viral genome sequences potentially. So any questions about any of that stuff? Feel free to type in the chat if you're on Zoom. Anyone in the class, all good? Kind of makes sense. Okay, so now what do we actually do with it? And I mentioned weeks like 5, 6, and 7 are kind of like a 3-week trilogy, essentially, of our ultimate goal being let's reconstruct the evolutionary tree of all these viruses. Let's figure out where is the most recent common ancestor. Let's figure out like what is the sequence of that ancestor. Let's figure out the evolutionary history of these things. So today is the first step towards that goal. We're going to talk about a problem known as phylogenetic inference. So here's an outline for today's lecture. I'm going to talk about mutations along a phylogeny. Hopefully folks remember the term phylogeny, but going back to, I think, week 1 when I introduced it, I think that's when it was. Phylogeny is just a fancy word for evolutionary tree. Don't worry about, I don't know like why we have a fancy word for it, but it is synonymous with evolutionary tree. So phylogenetics is just the study of evolutionary trees. Phylogenetic inference is just inferring an evolutionary tree. So we're going to review how we think about mutations following along an evolutionary tree. I'm going to introduce a concept known as continuous time Markov chain. So this is like something that if you've taken a machine learning class, you might have learned about, but if not, that's totally fine. We're going to teach it from scratch. Then we're going to talk about some different models of nucleotide evolution that we have. These are all going to be continuous time Markov chain models of evolution, which doesn't make sense to you yet, but it will hopefully 50 minutes from now, 40 minutes from now. And then we're going to end by talking about phylogenetic inference. So today's lecture is going to have a bit more math than usual, so bear with me, and if we end up not finishing, that's totally fine. We'll finish next time. But yeah, interject with questions as we go through, because this is, I think, a little bit more kind of like significant on the probability theory and that kind of thing. Okay, so questions before we begin? Concerns, comments? Everyone's buckled up, ready for a lot of math? Awesome, let's do it. Okay, so first let's talk about mutations along a phylogeny. This is pulled directly from the first lecture. Remember, we can think of a phylogeny as showing the kind of branching pattern of the evolutionary history of a bunch of sequences. We're at the top of this evolutionary tree, which is further back in time. We have some ancestral genome sequence. And then along the various branches of the tree, mutations occur, which are heritable and they get inherited all the way down the tree. So in present day, we're dealing with these sequences over here. In reality, we don't know the true evolutionary tree. So basically what we're working with is something like this. And even worse, we don't have access to those gaps over here. All we have access to in reality is just the actual genome sequences that we've assembled from these different samples. But now, as of last week, we can stick those gaps back in. We now learned about multiple sequence alignment. Perfect. We're one step closer to reconstructing the evolutionary tree. So this is where we left off last week. We have the multiple sequence alignment. Every one of those sequences in our alignment corresponds to one leaf in the evolutionary tree. And our goal is how do we infer the phylogeny that gave rise to these sequences? Okay. So we're going to start with a little bit of probability theory, talking about what's called a continuous time Markov chain. So first, I want to introduce the concept of a Markov chain. A Markov chain, it's a weighted graph. So you learned about graphs, you learned about weighted graphs. It's a so for example, and I have an example that I'll talk about in a little bit, but basically the nodes represent what states your entity or your system could be in at any given time. Oh, my internet connection's stable. So Zoom folks, if my internet cuts out, just like, I guess yell at me if there's any way. I guess if there's no internet, I won't be able to hear it, but that's okay. You'll figure it out. So yeah, so the nodes in this Markov chain represent the states that we can exist in. The edges, these are going to be directed edges the edges of a Markov chain represent transitions between the states. So our system is moving from one state into some other state. And the edge weights represent transition probabilities. So given that I'm at a given state, what is the probability of me transitioning to some specific other state? Okay, and I think it'll hit home a little bit better with this example Markov chain that we have. So here I have a Markov chain representing the weather in San Diego. The nodes of this Markov chain represent the states that the weather can be in. So maybe it could either be sunny, cloudy, or rainy. The edges represent transitions between the states. So basically, what is the transition from today being sunny to tomorrow also being sunny? Or today being sunny, but tomorrow's cloudy? Or today is sunny, tomorrow is rainy, etc. So for each of our states, we have, like if we have n states, we have n transitions for each state, basically going to myself or going to any of the other n-1 states. And then the edge weights represent transition probabilities. So basically, let's say if today is sunny, there's a 70% chance that tomorrow is going to be sunny as well. If today is sunny, there's a 20% chance that tomorrow's going to be cloudy, etc. One thing I want to emphasize is kind of going back to basic probability theory. If today is sunny, assuming that the world doesn't end tomorrow, which who knows, but if it doesn't, it should be one of these three states tomorrow. So the outgoing edges have to add up to 100%. So basically, for any one of these states, the outgoing edge probabilities have to add up to 100%. Otherwise, like, you disappear off the face of the earth. That's not good. Okay. So what is a Markov chain? Why am I calling it a Markov chain rather than just a weighted graph? So it has these properties, but then this term Markov has a special meaning as well. The term Markov has to do with what's called the Markov property, which states that a Markov chain is what we call memoryless. So I'm going to show you the mathematical definition of what memoryless property means, and then I'll give it in more kind of like colloquial English. So the mathematical definition of the Markov property, so of our Markov chain being memoryless, is that, so here, P is a probability, capital X is a random variable denoting the states that you can be in, lowercase x is like a specific state that you're in. So what this is saying is the probability that you happen to be in whatever state you were in at time I+1, given what state you were in time I -1, time I-2, all the way to time 0, so like the very first time step, is equal to the probability of being in whatever state you're in at time I+1, given what state that you were in at time I. So in English, what that means is where you go next only depends on where you currently are. So the probability of what state I'm in my next time step only depends on the probability of what state I'm currently in. It doesn't depend on the complete history of all the states that I've been in previously. So like even if it's been like a month of rain and then you have one sunny day, the probability of what the weather is going to be like tomorrow only depends on it was sunny today. It doesn't depend on the fact that it was a whole month of rain. That's a bad assumption, but it's a simplifying assumption. It makes the problem much simpler. Okay. So questions so far? Concerns? Anyone on Zoom? Okay, so we've defined what a Markov chain is. So what do we have to define in order to define a Markov chain? So you have to define the states. So what are the different states that my system can be in? So for San Diego weather, I said sunny, cloudy, rainy. Maybe you have drizzly as a state. Maybe you have, I don't know, foggy. I guess is foggy kind of cloudy? Not, I don't know. But what states you pick is one aspect of what you have to define for a Markov chain. Then once you have the states, I mentioned in that equation that there was a time 0. So what state are you going to be in at time 0, at like the beginning of the existence of your system? So like the beginning of the existence of the world, or at least when we started paying attention to the weather. We have to define what we call the initial state probability. So this is a vector that's telling us what is the probability that at the very first time point that I'm looking at, I was in any of the given states. So here I'm saying the probability that I was in state sunny on that very first day is 70%. The probability that that first day was cloudy is 20%. and the probability that the very first day of existence was rainy is 10%. So I have to say, what is the probability of what state I was in on the very first day? And then the last thing that we have to define is the transition matrix. So I mentioned we have those outgoing transition probabilities. So basically I have to define a matrix where each row is what state you're coming from, each column is what state you're going to, and the values of those cells is the probability of going from the row state to the column state. And just kind of reminding you what I said earlier, because wherever I am today, I have to go somewhere tomorrow, the sum of each row has to be 100%. It has to be 100% chance that given that I'm in a specific state today, I go somewhere. Okay. Questions? All good? Oh yeah, go for it, Yeah.
So we're just kind of defining the initial state probability is not calculated based on those edge weights.
Good question. So the question was, so are we just kind of defining these initial state probabilities, or is it calculated somehow from these transition rates? So the initial state probabilities have to just be pre-known somehow. So in this example, I just arbitrarily said, well, it's San Diego, so probably it's usually sunny. Sometimes it's cloudy and very rarely it's rainy. Whatever system that you're looking at, you would probably just look at like longitudinal data and see like, okay, let me look at the entire history of San Diego that I've ever known about. What portion of days were sunny versus rainy versus cloudy? And I can estimate it based on real data. In practice, sometimes people also just pick a uniform distribution for this. So if you don't have any additional information, maybe you'll just say 1/3, 1/3, 1/3, just because you don't know any better. Yeah. But yeah, you do have to know it. In statistics, we call this a priori. In advance, you have to know what is that probability vector, whether you know exactly what it is from real world estimates or you just guess uniform distribution or something. Yeah, great question. Awesome. Other questions? Yeah, go for it.
Guess every node to every other node, even if it's like a really low probability of like one node to another.
Good question. So the question was, is it guaranteed that in a Markov chain, we have an edge from every node to every node, even if it's a really low probability? In general, when we define a Markov chain from like a mathematical theory perspective, yeah, we have every single possible pairwise edge. In practice, if some of those states are impossible, so like let's say it's impossible to go from sunny directly to rainy, you still would have that edge in your Markov chain. It's just that you would have a probability of 0. But yeah, in theory, we have all the edges, but in implementation, sometimes you'll like throw away some of the edges if it's actually impossible. Yeah, great question. Okay, so now that we've talked about Markov chains, what's a key limitation of this specific example that I've given you? So I've given you a Markov chain that represents the weather, and I've talked about, okay, we have a way of jumping from what weather it is today to what weather it is tomorrow and kind of jumping day by day. Does anyone have any like problems with this model that I've come up with? Is it a good model of the weather? No, how come? But I made it.
Well, the weather is affected by previous days.
Oh, so weather is affected by previous days, but for my purposes, I'm keeping my model simple. So I'm acknowledging the Markov assumption is a bad assumption because I'm throwing away past history, but I'm going to stick with it because it's easier. But any other thoughts? Exactly. So the answer was in a given day, it can cycle between states within that same day. So like this you all took CSE 21, I think. It's like discrete math. Discrete means integer, like counting. This is a discrete Markov chain. I'm saying today is one weather. Tomorrow is another weather. The next day is another weather. That's not a very realistic model of weather. Weather is kind of like continuously transitioning, like it'll start sunny, and then maybe clouds come in, and then maybe the clouds slowly move away, and then like the clouds come back, and then it starts raining, then it stops raining, etc. So discrete time is not a very good way of describing this specific real world phenomenon. What do? We can introduce what are called continuous time Markov chains. So In the previous example, time was discrete. We were using 1 day steps. Maybe it's 1 hour, maybe it's 1 minute, maybe it's 1 second, but they're still integer discrete time steps. But in reality, this phenomenon is a continuous time phenomenon. So a continuous time Markov chain, very easy definition. It's just a Markov chain, but instead of having discrete time steps, we're using continuous time. So time progresses continuously. What that means in terms of the math, you all don't have to worry about. If you ever take, is it like CSE 150A or 151A? I don't know. There's like some CSE class that talks about Markov chains. Maybe it's 150B. If you do more work in this space, you'll have to learn how that messes up the math. For our purposes, the kind of 1 sentence summary you can just remember is probabilities deal with discrete events, a coin flip, the roll of a die, some discrete event that I took. Since this is continuous time, these transition probabilities are replaced with what we call transition rates. We're kind of like continuously transitioning from this state to all the states simultaneously, and just randomly one of them ends up succeeding. So what that implies for the math, don't worry about, but that's just kind of like, imagine we're lessening the time step infinitely small until the derivative, I guess not derivative, like until we hit 0 kind of time step, infinitely small. Okay. Cool. Okay, so that is my brief summary of what a continuous time Markov chain is. So in summary, we have states, we have transitions between those states, and those transitions happen continuously. Okay. Okay, so this next section, I'm going to introduce how we can use continuous time Markov chains to model nucleotide evolution. So if we model nucleotide evolution as a Markov chain, let's define those different properties that I said we have to define. What are the states? Well, a given nucleotide is either going to be A, C, G, or T. And then the initial state probabilities, what we're going to use is going to depend on the specific model, but like maybe I just say 25% chance for all 4 of them, or maybe I look at the entire genome and I see like, 60% of it is A, maybe 60% A is really high. Maybe 30% is A, 30% is T, 20% is C, 20% is G. So maybe I'll use those as my initial probabilities. But somehow I come up with what I think the initial state probabilities are. The transition rates, again, also depend on the specific model. So like, what is the rate at which we go mutate from A to T, or mutate from A to C, or mutate from A to G, etc. I'll have to define what are those transition probabilities. The transition rates, because this is continuous time. So I can think of it as filling in a matrix like this. Okay. The last thing that I'm going to mention is models of nucleotide evolution. Usually we use what are called time reversible models. So a time reversible model means that the transition from one nucleotide, let's say X to Y, is the same rate as transitioning from Y to X. So we're assuming that it's kind of like mirrored, like it's the same rate from X to Y as it is from Y to X. This makes the math a little bit easier, but it's not necessarily realistic. So there do also exist non-time reversible models, but the ones that we're going to talk about in this class are time reversible. Yeah, go for it.
But like, there's a lot of like a common mutation is like mutating from like C to G.
Ooh. So isn't it, so the question was like there's certain common mutations, like, so C to G, not as much. A to G is one. C to T is a common one. So there are some mutations that are more common than others. So hold on to that thought. We're going to rapid fire a bunch of models in a second, and that's going to come up. But last thing about this slide is because it's time reversible, so hopefully intuitively that tells you that things below the diagonal I don't have to define, because A to C is equivalent to C to A. So I don't have to separately define C to A. What's not as obvious, because we're dealing with rates, but just trust me, and then you can learn about it later, is we also don't have to define the diagonal. And the kind of analogy that I'll use is with respect to, go back to discrete time. Remember I said that the sum of the rows has to add up to 1. It turns out that if we know the average mutation rate, there's similar fancy math that we can do equivalent-ish to sum of a row equals 1. We can figure out this rate based on these 3 rates and based on the average rate of the entire system. So just trust me when I say we also don't have to calculate or like have values for the diagonal either. So in short, if we're using a time reversible model of nucleotide evolution, we only have to figure out these 6 transition rates, and then all the other ones are defined. Okay. So I'm about to go on a rapid fire of a bunch of different models of evolution. I guess before I do that, any questions, comments? Okay. So before I go on that rapid fire, I want to emphasize, don't focus on like memorizing what all the terms of these different models are. You're never gonna, I mean, I have to know them nowadays, but like, you're never gonna have to just like off the top of your head memorize like what are the differences between these models. I think the main thing I want you to think about is what are the model assumptions that each of them are using and how do they compare against each other in terms of like how flexible or inflexible they are. So compare them, just like buckle up and go for the ride, but don't focus too hard on like trying to memorize everything. Okay, so hands and seats are in the roller coaster. Let's go. Okay, so the simplest model that we can come up with, this is called the Jukes-Cantor model of 1969. And for all these models, I'm going to kind of highlight how do we do the base frequencies for that initial state probability, and also how do we handle the transition rates. So the base frequencies, it just assumes are all the same. So 25, 25, 25, 25, easy peasy. And it also assumes all the possible mutations are also equivalent. So there's just one global mutation rate that is used for all of these different transitions. And we just assume all the probabilities are 25%. So this is the simplest that we could do. We just have a mutation rate, and then everything else is just kind of neutral. Okay. So as you said a second ago, wait a minute, some mutations are more likely than others. So has anyone taken OChem? And you survived. Good job. I didn't survive that much. I'm just kidding. So has anyone taken 140C, or I guess now it's called 40C? No? Okay. So, or maybe metabolic biochem. Has anyone taken that? Yeah. So have they talked about like, did they talk about like the nucleotides at all? Like the nucleic acids? Ooh. Exactly. So for reasons that we don't have to worry about in this class, but just as the background for why we have these more sophisticated models, it turns out that because of organic chemistry reasons that you can look up on Wikipedia, or you can take a hard class later if you want, because of the organic chemistry structure of the nucleic acids, like of the bases, there's some mutations are easier than other mutations. So specifically, there's some terms that we have called purines. So the purines are A and G. The pyrimidines are C and T. And because of shared organic chemistry properties between the purines and shared organic chemistry properties between the pyrimidines, it's easy to go within those classes, which we call a transition, than it is to go across those classes, which we call a transversion. So in 1980, a researcher named Kimura came up with the Kimura 1980 model. All these models are going to be like someone's name and then a year. We like to name our stuff after ourselves. It's just what we do. So the Kimura 1980 model says, okay, well, I'm going to keep using this simplified equal probabilities thing from the JC69 model, but now let me have 2 different mutation rates. Let me have the transition mutation rate, which is purine to purine or pyrimidine to pyrimidine. And let me have the transversion one, which is purine to pyrimidine or pyrimidine to purine. So we have to define 2 transition rates on this matrix. Okay. So in 1981, Kimura wasn't done yet. He was like, actually, I want to be a little bit more kind of nitpicky. So we're going to have transitions. And then now this is outside of my knowledge of like organic chemistry. I don't know what weak strong properties are. I don't know what amino keto properties are, but if you've taken chemistry stuff, you might know these. But basically some property of the amino acids that's, oh, do you know it? Go for it. Okay. Ooh, so it has to do with how many hydrogen bonds are between them. Okay, awesome. Good catch. I was testing you. I knew that the whole time. So basically, we're now distinguishing within the transversions. We have 2 different types of transversions that we're distinguishing between. So now we have 3 mutation rates, 1 for transitions, 1 for transversions that are conserving weak strong properties, one for transversions that are conserving amino keto properties. So basically what that means for us as the bioinformatician is now there's 3 rates in my matrix that I have to keep into account. Okay, so in 1981, a guy named Joe Felsenstein was like, wait, let me go back and let me take another look at that JC69 model. So in this model, the Felsenstein 1981 model, we're assuming that the rates are all equal. Again, we're going back to that simple, let's just assume there's one global mutation rate. But now, instead of assuming all four initial state probabilities are equal, he lets you pick whatever value you want for these. So you now have like 3 degrees of freedom on this front because, again, they have to add up to 100% because you have to have one of the 4 probabilities. But at the very least, they could all be different values if you want. Okay, so simple here, but now flexible with the probabilities. Okay, folks are still with me. Okay, in 1985, Hasegawa, Kishino, and Yano came up with a model that has this flexibility with the probabilities again. But then just like the Kimura 1980 model is also able to distinguish between transition rates and transversion rates. So there's a question in the chat. Wouldn't the frequency of A equal C and the same for G and T? Oh, you mean A and T and G and C. So great question. So the probability of A and the probability of T are usually almost equal, but there could be variation a little bit, especially when we're dealing with like single-stranded genomes. So things like an RNA virus, you could get deviation from the complementary bases. So usually they'll still be close, but it could deviate a little bit. It won't be like exactly the same number. Great question. But hold on to that thought, because I have a slide coming up next that's going to touch on that. But hold on to that thought. But yeah, so we have flexibility on all 4 of these, and then transitions and transversions, just like the Kimura 1980 model. Okay. So the next one, Tamura had exactly the same idea that you did, where he was like, wait a minute, we can use Chargaff's principles and say probability of A is equal to probability of T, probability of C is equal to probability of G. So this one is basically taking the Kimura 1980 transition transversion thing and is a little bit more flexible in terms of these probabilities, but it is forcing you to have the same A to T, same C to G. Awesome. Yeah, you go for it. Sorry.
You were like creating a system that like does this. When like one of the things you want to like clarify is like if it's like a single stranded or a double stranded. If it's like if it's double stranded, it's probably this, and if it's not double stranded, there's probably and if it's like single stranded, there's probably variation. On like A to T won't be the same.
Good question. So the question, just to kind of repeat it, so basically, if I'm using a system like this, should I take into account like, is the thing that I'm analyzing single-stranded or double-stranded, or like what assumptions can I make? So in terms, I'll kind of slightly distinguish between, there's the people coming up with the models, and then there's the biologist using the model to analyze real data sets. And just like with bioinformatics tools, where like we have to pick, like, I don't know if any of the read mappers that we used were splice aware, and the TA is not here. Or maybe Kira's on the chat. Oh, was it?
HiSat.
Oh, HiSat was, yeah, perfect. So we did use HiSat. So some read mappers assume no splicing, and they assume, okay, this read is going to almost perfectly match somewhere. Versus if you imagine if I have RNA-seq and I've spliced out an intron, if I have a read from an RNA-seq experiment that spans that intron junction site, and we'll talk more about RNA-seq in week 9, but basically if I have a read that spans an intron splice site, when I map it to the reference genome, I'm going to get 2 halves of this read that are like kilobases apart potentially. So in the same way that a biologist needs to think about like, oh, I'm analyzing RNA data, so I should use the right tools that assume the right things. Same thing here. When you're picking what model you want to assume, you should use a model that's realistic for, okay, is it a DNA or an RNA genome? Is it the whole genome, or is it a specific gene or whatever? So like, yeah, so like basically you'll have to consider all of those features when you're picking what model you want to use. But for now, when we're defining the models, we just define the rules, and it's kind of up to the biologist to pick the right one. Yeah, great question. Awesome. Any other questions? Feel free. Okay, we're almost done, I swear. So the next model, this is the second to last one that I'm going to talk about. This is called the Tamura-Ney 1993 model. So we allow all 4 of these probabilities to vary. And this one has 3 different like mutation rates that it distinguishes between. These are different from the K81 3 different mutation rates. That one was transitions and then 2 different types of transversions. This one, we have 2 different types of transitions. We have A to G transition, C to T transition. These are 2 separate rates. And then we're lumping all transversions in the same bucket. Okay. And then for reasons you don't have to worry about this week, but you will. This is week 6. So I think you talk about it week 8, maybe, or week 7. I forgot what week. So the TN93 model is the most generalized model that these model parameters can be inferred directly from a multiple sequence alignment. Why that's the case, don't worry about it. I'm also never going to test it on an exam, but just for your own information, because we're going to use a tool later on that calculates Tamura 93 or TN93 distances for a later project or like for a later lab. That's just kind of a heads up, is that this is the most generalized model that we can infer the model parameters directly from a multiple sequence alignment without needing the evolutionary tree. Okay. So that's second to last. The very last one that we're going to talk about is called the generalized time reversible model. So all of the bases can vary, and all of these transition rates can vary. And in general, this is the most generalized model that people typically use. If in reality, one of those other models is more appropriate, this should converge on it, because if you think about it, all those other models are what we call instantiations of this model. So if you think the Jukes-Cantor model where all these are the same and all these are the same is actually the real one, a real data set that has those properties, you're going to end up with inferring 4 probabilities that are very close to 25%, and you're going to end up inferring a bunch of transition rates that are almost equal. So if any of the other models was technically correct, this should also converge to those same model parameters. If it doesn't, those models probably weren't correct. But the distinction is the more model parameters you have, the slower it is to learn them. So it's kind of like a performance trade-off between more flexible model equals more runtime and memory typically, less flexible model equals much faster and much less resource intensive. So usually you want to use the simplest model that's appropriate for whatever context you're doing. There's a question, why are the transitions put into 2 groups for this model? Good question that I'm not sure. So I forgot what, so I can, if you want, feel free to let me know and I can send you a link to the paper for the TN90. This is about the TN93 model. I think they were studying like mitochondrial DNA or like some specific biological property. And this setup was motivated by like the biological constraints of like whatever they were studying. So I don't remember off the top of my head, but if you want, I can send you the paper. And yeah, it's a good paper. Awesome. Yeah, go for it. Go for it.
To discover like a different one, like the ones that they have for like your specific set of like data.
Yeah. So couldn't you oh yeah, go for it.
You're trying to use data and like it's not matching all the models, so you just use like a general one. And you find that like there is a pattern, but it's like different than like, the patterns you've seen.
Yeah, good question. So I guess I'll kind of like expand your question of like, what if none of these models quite fit your data? So like in general, if you assume that time reversible assumption is okay, this is the most generalized that you can theoretically get. So this should pick up on anything. If even this doesn't work, then you would have to say like, oh, hey, all the models that I have at my disposal suck. Uh-oh, I have to make my own, which then you get a paper out of it because you came up with your own model. So again, a lot of people have beef with me for some reason because of COVID origins research. And this was one of the issues with COVID origins research was in the early sequences, we didn't want to assume time reversibility because we actually had like clear signal of there's only like a couple sequences and we can see like the directionality of mutations. So when we were inferring the evolutionary tree to then figure out what is the most likely common ancestor of all of these, we had to come up with like a novel model that didn't assume time reversibility, and we had to come up with all the mathematical derivations of it and all that kind of headache stuff. So usually you want to use one of these ones out of the box just because the math is kind of hard. But sometimes these fail. They're not appropriate for whatever context you have. And you have to like, convince the reader, here is exactly why this model is insufficient. And here is exactly how it breaks. Here's a new model that we developed that is more appropriate. And here's the proof of how it's actually a more appropriate fit for the data set. And then that's a lot of work. And then you get a Nature paper out of it. And then yay, happy. Or science or something. Yeah.
Do we just use single nucleotide for the states, or do we put like, see, like how does K-mer work for like?
Good question. So the question was, do we always just use single nucleotides for the states, or can we get more complex? So you said K-mers, we could also do codons. We usually, if we're doing DNA sequences, it's usually either single nucleotides or codons, like 3 nucleotide chunks. So there's a separate class of evolutionary models called codon models. And those, instead of having a 4x4 matrix, you're going to have 64x64 matrix of all of the different possible codons. Yeah. And those, they're more appropriate for like, so a codon model, why do we care about codons biologically? Okay, well, codons are how we do translation. So a codon model would be appropriate if you know in advance, I'm only looking at protein coding sequences. If I'm looking at the whole genome, it might not be appropriate because like the intergenic parts of the genome, there's no reason why they would necessarily like adhere to a codon model. So it introduces 64x64 model parameters that you then have to learn, which kind of muddies up the water. So if you know in advance, I'm only looking at coding regions of a genome, and I know that they're in frame, absolutely. We have a whole class of codon models for that. Great question. And we also have amino acid models that I'm not talking about today, but single letter amino acids. Awesome. Other questions, concerns? So we have 7 minutes left. I think we can get through most of it. So that's good. Okay. So this describes the evolution of a single position of the genome sequence. However, we're not talking about phylogenetic inference from one letter. We're talking about phylogenetic inference from a multiple sequence alignment of entire genomes or entire genes or whatever. So how do we deal with heterogeneous mutation rates? Well, first of all, what does that even mean? Well, all those substitution models I just talked about, they're for a single position or a single site of the genome. But yeah, how do I expand it to handle entire genome sequences? The most simple assumption that I could make, and I'm going to emphasize simple, not correct, simple. The most simple assumption I can make is I can assume that all of the sites of my genome are in the statistics role what we call IID. They're independent and identically distributed. So remember, independent means whatever happens here has no impact on whatever happens here and vice versa. Identically distributed means exact same model parameters. So I have a bunch of identical processes with the exact same model parameters, and they don't impact each other whatsoever. You just mentioned codons. Why did you mention codons?
Oh, I thought like, because I know like people do Markov chains on n-grams, and I thought that just came right from mathematics.
So anagrams are important. What about like where so with respect to translation, what do you think might happen? Like why might codons be important?
Or just mid-sense and then mess up all the protein products.
Exactly. So the answer was it messes like if we have a missense or a nonsense mutation, we can completely like screw up the protein product. So assuming these sites are independent is a very bad assumption for coding regions, because if I accidentally cause a nonsense mutation in this site, I've destroyed the entire protein product. So identically distributed, or no, sorry, independent is a very bad assumption because the sites are not actually independent necessarily. They have very significant constraints if I'm in a coding region. There's also coding versus non-coding regions of the genome. Coding regions have a lot more what we call selection pressure. If you mutate this part of the genome and it breaks the protein, the virus or the organism or whatever dies. Versus if I mutate something in the intergenic region, it might be more flexible towards mutations because, hey, this thing is still going to survive if it's like just kind of physical padding. So identically distributed also doesn't make sense because some parts are going to mutate more than others. Yeah, go for it.
On like top of that, is there also like some sections of the genome might be considered like coding regions, but they're completely like methylated, so I think they can just be accessed.
Yeah. So some parts, so you mentioned methylation. So with epigenetics, now we have like some parts of the genome might be methylated or acetylated. And like if we take into account, like we as bioinformaticians like thinking of DNA as like one long string, and we have like 23 long strings in our nucleus. But actually, it's like this wound up, really weird thing. And transcription is not just we are transcribing this part of the genome. It's like, oh, this really crazy thing gets some epigenetic modification that like physically opens it, and now this part of the genome is exposed, and now we have this downstream stuff transcribing from that. So yeah, epigenetics also has a big impact on like, we can't just assume these are all identical. And empirically, I can actually look at, so here, this is a plot. So this is of SARS-CoV-2 genomes. Horizontal axis is the SARS-CoV-2 genome. Vertical axis is Shannon entropy. So entropy, if you've taken 100, you know what Shannon entropy is. If you haven't taken 100, think of it as like a measure of disorder. So the higher this entropy, the more variation there is at this site. The lower the entropy, the more the same everything is. So you can kind of imagine interpreting this plot as like vertical means like more variation is sustained in different genomes at this position. So you'll notice like the spike protein of SARS-CoV-2 has a lot of mutations because it does a lot of like immune evasion versus like some of the other parts of the genome that maybe are more important for like basic virus survival or functionality. They're not allowed to mutate as frequently. So looking at this plot just right off the top of your head, clearly IID is a terrible assumption for this. So one improvement is to assume some type of mutation rate heterogeneity across sites. So basically, instead of saying every site has the exact same mutation rates, let's imagine that some sites have more mutation, some sites have less. And usually the models that we use assume each site has the same relative rates. So like A to C, A to G, A to T have the same proportions, but we can use some distribution to say what is the expected mutation rate, what is the mean mutation rate at any of those sites. So like maybe one site has a way higher mean, but then within it, each of those mutations has like the same proportional rates, versus another site, we have a lower mean mutation rate, but the different mutation rates are proportionally the same. So kind of like scaling it up or down according to some model. So in practice, people use the gamma distribution very frequently. So you assume a gamma distributed mutation rate distribution with some mean, that's your overall mean distribution. But then the actual distribution, the actual mutation rate at each site is sampled from this gamma distribution that you fit. Another improvement that we could do is allow some positions to be what we call invariant. So allow some of them to just have a mutation rate of 0 and not try to learn the mutation rates from our substitution models, not try to learn this heterogeneity gamma distribution, just ignore them from all the downstream calculation, just say this site is invariant, don't use this for any downstream model interpretation. So yeah, this prevents invariant sites from screwing up that mutation rate distribution with outliers of 0. So we're at 50 after. I'm going to stop here. But the last thing that we're going to talk about is now that we've learned all these different models of nucleotide evolution, how do we actually use them to do phylogenetic inference, to actually infer an evolutionary tree from these multiple sequence alignment? Cool. So any questions before I end the slides or end the Zoom call or anything? Anyone on Zoom, feel free to type in the chat. Anyone in the room, questions, concerns? No? Awesome. Well, thanks so much, folks. I guess good luck on the lab, and I will see you all on Wednesday. Thanks. Bye, everybody.